Have you ever stopped to think about the tiny molecules working behind the scenes in your body? One of the most crucial yet least talked about is urea. You may not know its name, however, it's constantly at work, helping detoxify and balance your body in ways you may never have imagined. It's involved in processes that keeps you healthy, from filtering toxins to supporting your organs.
Urea isn't just a random molecule. It is essential to your health. It keeps your body healthy and safe almost every day. Yet, most people don't even know it's in there.
And get this, urea isn't just a bodyguard inside your body. It's also a powerhouse outside your body.
From fertilizers that help crops grow to skincare products that hydrate your skin, and even chemicals in plastics and resins, urea is everywhere.
So the question is, do you know the full story behind this small but unique molecule? You don't? Well, let's get on with the video.
Good day everyone. Today we're gonna talk about the urea cycle. So the first thing that you should know about the urea cycle is that it is also known as the Krebs-Henseleit cycle or the ornithine cycle. It is a metabolic pathway in the liver that detoxifies ammonia. Ammonia, which you may not know, is a toxic byproduct of protein metabolism. So in this cycle, that ammonia is converted into urea, which is less toxic. So the key difference between ammonia and urea is that ammonia is toxic and water insoluble, while urea is non-toxic and water soluble, which is why urea can be excreted through the urine, while ammonia can't. So here you can see Hans Krebs and Kurt Henseleit. They are the primary people responsible for discovering urea cycle. Urea cycle is the first metabolic cycle described by them in 1932, hence its name. So the urea cycle primarily occurs in two main locations within the body, in the liver and the kidneys. Here in the liver, it occurs primarily in the hepatocytes, specifically in the mitochondria and the cytoplasm. This organ is crucial for processing and detoxifying substances. Here, the liver converts ammonia, a toxic byproduct of protein metabolism, by combining it with carbon dioxide to produce urea, which is less toxic. It is then transported to the kidneys. In the kidneys play a vital role in excreting urea. It filters out the urea, which is then excreted from the body in urine. So now we're gonna talk about the purpose of the urea cycle and discover why it's so important. So the first reason why the urea cycle is important is because it detoxifies ammonia. When proteins are metabolized, they release amino acids, which contain nitrogen. This nitrogen, if not managed properly, forms ammonia, a highly toxic compound. Ammonia is particularly harmful to the central nervous system because even small amounts can disrupt cellular processes, including the Krebs cycle and neurotransmitter production. To prevent ammonia accumulation, the liver converts it into urea through the urea cycle. This process protects the brain and other organs from ammonia toxicity by transforming it into a form that can be safely transported and excreted by the body. Next, it regulates nitrogen levels in the body. By converting excess nitrogen into urea, the urea cycle helps keep the body's nitrogen levels balanced. This is important for protein metabolism and overall health. Nitrogen is crucial for creating amino acids, nucleotides, and other cellular components. However, excess nitrogen from the breakdown of proteins and nucleic acids can disrupt cellular balance, leading to harmful conditions like hyperammonemia. The urea cycle allows the liver to convert excess nitrogen into urea. Lastly, the urea cycle is very important because it prepares for excretion. It is a way for the body to safely remove nitrogen waste and excess nitrogen. Urea produced in the cycle is transported from the liver to the kidneys, where it is then filtered out and excreted through urine. This process not only helps eliminate nitrogenous waste, but also prevents the buildup of toxic compounds in the blood. So before we get into the urea cycle itself, we must first understand how protein is metabolized. So now, let's get into the process of protein metabolism. Before the protein gets into the liver, it first gets metabolized and goes through two processes, the transamination process and oxidative deamination. So here in transamination, an amino group from an amino acid is transferred to an alpha keto acid. This process allows the amino acid to be converted into a different keto acid, while the original keto acid becomes a new amino acid. This reaction is catalyzed by enzymes such as transaminases or aminotransferases, where the two important transaminases for the urea cycle are the alanine transaminase or ALT, which typically transfers an amino group from alanine to alpha-ketoglutarate, forming pyruvate and glutamate, and aspartate transaminase or AST, which transfers an amino group from aspartate to alpha-ketoglutarate, forming oxaloacetate and glutamate. Next, let's move on to oxidative deamination. This reaction focuses on removing the amino group from glutamate, which was produced during transamination. In this process, glutamate is broken down to release ammonia and alpha-ketoglutarate. The ammonia released in this process enters the urea cycle in the liver, where it's detoxified and excreted from the body. This reaction is catalyzed by an enzyme called glutamate dehydrogenase and occurs primarily in the liver. So now that we know protein metabolism, or how ammonia is formed through transamination and oxidative deamination, let's now get onto the urea cycle itself. So, the urea cycle primarily occurs in the liver, specifically in the mitochondria and the cytosol of the cytoplasm. So the first thing that happens here is the formation of carbamyl phosphate. In the first step, ammonia produced from amino acid breakdown combines with carbon dioxide to form carbamyl phosphate. This reaction requires ATP for energy and is catalyzed by the enzyme carbamyl phosphate synthetase 1 or CPS1. In the next step, carbamyl phosphate combines with ornithine to form citrulline. This reaction is catalyzed by the enzyme ornithine transcarbamylase. Citrulline is then transported out of the mitochondria and into the cytosol of the cytoplasm, where the next step occurs. In the cytoplasm, citrulline combines with aspartate to form argininosuccinate. This step requires another ATP molecule and is catalyzed by the enzyme argininosuccinate synthetase. Next, argininosuccinate is cleaved by the enzyme argininosuccinase to form arginine. This step is important because arginine now carries the nitrogen that will be removed as urea in the final step. In the final step, arginine is hydrolyzed by the enzyme arginase to produce urea and ornithine. Ornithine is then transported back into the mitochondria to participate in another round of the urea cycle. Urea, now as a form of nitrogen, is released into the bloodstream and filtered out by the kidneys for excretion. Next, to add on to that, argininosuccinate can also be cleaved by the enzyme argininosuccinase to form fumarate. Fumarate is an intermediate of the citric acid cycle and can be recycled to help produce more energy. Fumarate can then also be converted to malate and then oxaloacetate. That oxaloacetate can also be used for gluconeogenesis, or it can be used to produce more aspartate through the enzyme aspartate aminotransferase, or AST, which was also found in the process of transamination. Now, this enzyme converts oxaloacetate into aspartate by taking an amino group from glutamate and transferring it to the oxaloacetate to make aspartate. This process is what you'd call the transamination process. So really, the aspartate is just oxaloacetate plus an amino group. So now, as you can see, this cycle can continue if you keep having aspartate in it. And in fact, you need high amounts of aspartate in order to have the urea cycle operate. So, you need that aspartate for that argininosuccinate synthetase reaction.
So now that we know the cycle, let's take a look at its ADP usage. Which parts of the urea cycle actually use ADP and how much? The first part of the urea cycle that uses ADP is the reaction with carbamoyl phosphate synthase 1 or CPS1. It requires 2 ADP molecules, and since it needs 2 ADP molecules, it's an irreversible reaction. So, once the hepatocyte produces carbamoyl phosphate, it cannot go back to ammonia. Here's a breakdown of how the ADP is used. So first off, the first ADP activates bicarbonate. Bicarbonate then reacts with ADP to form carboxyphosphate and ADP. Then, ammonia reacts with carboxyphosphate to form carbamate. This carbamate is then phosphorylized using a second ADP to form carbamoyl phosphate. Summing all these processes, we can see that CPS1 needs 2 ADP to make carbamoyl phosphate. Now, let's take a look at how N-acetylglutamate regulates the urea cycle. The enzyme carbamoyl phosphate synthetase 1, or CPS1, plays a key role in the urea cycle. Since we all know by now that CPS1 is an irreversible enzyme, it needs tight regulation. This is where N-acetylglutamate comes in as its activator. N-acetylglutamate is synthesized from acetyl-CoA and glutamate, with the enzyme N-acetylglutamate synthase responsible for its formation. So, what regulates N-acetylglutamate synthase? There are 2 main factors. 1. Arginine. 2. Starvation. Arginine is a natural activator of N-acetylglutamate synthase. When arginine levels are high, it signals the need for the urea cycle to process excess nitrogen. Starvation is another regulator. During starvation, the body breaks down proteins for energy, leading to a higher nitrogen load. To handle this, the body needs to excrete more nitrogenous waste, which then activates the urea cycle. So, through N-acetylglutamate, CPS1 activity is regulated, helping the body efficiently process and excrete nitrogen. The other enzyme that requires ATP is argininosuccinate synthetase. This reaction also uses the equivalent of 2 ADP molecules, even though it's often said to only use 1 because ADP here is converted to AMP. Here's how that works. First, ADP reacts with citrulline to form an activated intermediate called citrulyl AMP, which releases pyrophosphate. Now, citrulyl AMP is a more reactive intermediate, allowing aspartate to join, which then releases AMP and forms argininosuccinate. And here's the key part. The hydrolysis of pyrophosphate into 2 inorganic phosphates effectively doubles the energy cost, make it equivalent to using 2 ADP molecules. So, where does ammonia come from? Let's break down the main sources. First, proteins. When proteins undergo proteolysis, they're broken down into amino acids, which contain nitrogenous compounds. This breakdown releases ammonia as a byproduct. Another source is the glutaminase reaction. When you take glutamine, glutaminase converts it to glutamate, releasing ammonia. This mainly happens in the kidneys and liver, where it helps with acid-base balance and ammonia disposal. Next, there's the glutamate dehydrogenase reaction, or GDH. GDH deaminates glutamate to form alpha-ketoglutarate, releasing ammonia in the process. This reaction, which occurs in the liver and kidney mitochondria, helps dispose of nitrogen from amino acids. Then, we have amino acid and amine oxidases. These enzymes remove amino groups from amino acids and amines, producing ammonia. They mostly operate in the liver and kidneys. Another source is purine and pyrimidine metabolism. Breaking down nucleic acids releases ammonia as a byproduct, especially in high turnover tissues like the liver. Lastly, intestinal bacteria. Gut bacteria produce ammonia during protein digestion, and this ammonia can enter the bloodstream. In cases like bowel perforation, the ammonia load may increase because there is more direct absorption into the blood. Aside from these sources, there are also factors influencing the urea cycle. First up, protein intake. A high protein diet increases amino acid levels, which leads to more ammonia production through deamination. This higher nitrogen load stimulates urea cycle enzyme activity to help manage it effectively. Next, there's the metabolic state. For example, during starvation or prolonged fasting, the body starts breaking down proteins for energy. This increases ammonia levels, and if protein breakdown exceeds the urea cycle's capacity, the liver might struggle to detoxify the excess ammonia. Liver function is also crucial. In liver diseases like cirrhosis, the efficiency of the urea cycle decreases. This can result in elevated blood ammonia levels, especially with increased protein intake. Another factor is substrate availability. High concentrations of certain amino acids, like glutamate and aspartate, stimulate the synthesis of N-acetylglutamic acid. This molecule activates carboxyl phosphate synthesis 1, or CPS1, which enhances urea production. Finally, the gut microbiota. Intestinal bacteria produce ammonia, particularly in certain gastrointestinal disorders like a perforated bowel. This can lead to increased ammonia levels in the bloodstream.
So, as we all know by now, the urea cycle is a critical metabolic pathway in the liver that converts toxic ammonia into urea, which is then excreted from the body to detoxify ammonia and maintain nitrogen balance in the body. Urea cycle disorders, or UCDs, have significant clinical relevance due to their impact on ammonia metabolism and the potential for severe health consequences. Here are some. So first, we have hyperammonemia and its consequences. Urea cycle disorders, or UCDs, are genetic conditions that result from deficiencies in one or more enzymes involved in the urea cycle. These disorders lead to hyperammonemia, which are characterized by elevated levels of ammonia in the blood, which is highly toxic, especially to the central nervous system. Symptoms can range from mild to severe if not treated promptly. Second, we have the types of urea cycle disorders. There are several types of UCDs. These include: first, we have the N-acetylglutamate synthase, or NAGS deficiency. It is a rare genetic disorder that affects the urea cycle, leading to the accumulation of ammonia in the blood. This condition is caused by a deficiency of the enzyme N-acetylglutamate synthase, which is essential for synthesizing N-acetylglutamate, or NAG, an important activator of carboxyl phosphate synthetase 1, or CPS1, the first enzyme in the urea cycle. NAG's deficiency impairs CPS1 activity due to its lack of a sexual activator, leading to similar symptoms as CPS1 deficiency, including severe hyperammonemia. This autosomal recessive disorder is less common but clinically relevant. Next, we have carboxyl phosphate synthetase 1, or CPS1 deficiency. CPS1 deficiency is a rare and severe metabolic disorder that affects the urea cycle, leading to the accumulation of ammonia in the blood due to a deficiency of the CPS1 enzyme, which is crucial for converting ammonia and bicarbonate into carbamoyl phosphate. It is inherited with autosomal recessive inheritance, meaning both parents must carry a mutated gene for a child to be affected. This causes severe hyperammonemia and is one of the most severe forms. The severity of symptoms often correlates with how proximal the enzyme deficiency is within the cycle. Deficiencies earlier in the cycle typically result in a more severe hyperammonemia. Ornithine transcarbamylase deficiency, or OTC deficiency. These are the most common UCDs, leading to increased levels of carbamoyl, phosphate, and orotic acid, and it is inherited in an X-linked recessive manner. It leads to elevated ammonia levels and orotic aciduria due to the accumulation of carbamoyl phosphate, which is shunted in the pyrimidine synthesis. Symptoms can range from mild to severe, with affected males often presenting with hyperammonemic crisis shortly after birth. Female carriers may experience milder symptoms or postpartum hyperammonemia. Next up, we have argininosuccinate synthetase, or ASS deficiency, which is commonly called acetyleninemia type 1. ASS deficiency is a genetic disorder that affects the urea cycle that also leads to the accumulation of ammonia in the blood. This condition is caused by a deficiency or absence of the enzyme argininosuccinate synthetase, which plays a crucial role in converting citrulline and aspartate into argininosuccinate, which results in elevated citrulline levels. It is also inherited with autosomal recessive inheritance. Next in the cycle is argininosuccinate lyase, or ASL deficiency, which is also known as argininosuccinic aciduria. It is a rare genetic disorder that disrupts the urea cycle due to a deficiency of the enzyme argininosuccinate lyase. This enzyme is crucial for the conversion of argininosuccinate into arginine and fumarate. Its deficiency leads to the accumulation of ammonia in the blood, which is toxic. Patients may have elevated argininosuccinate levels and can exhibit developmental delays, seizures, and behavioral issues. Next, we have arginase 1 or ARG1 deficiency, which is also known as the argininemia deficiency. It is a rare inherited metabolic disorder that affects the urea cycle, specifically the final step of nitrogen removal from the body. This condition is characterized by a deficiency of the enzyme arginase, which leads to the accumulation of arginine and ammonia in the blood. This autosomal recessive disorder is characterized by a later onset of symptoms. While it does not typically present with acute hyperammonemia, it can lead to progressive neurological decline and spastic diplegia. Number 3 is diagnosis and management. Diagnosis of UCDs typically involves serum amino acid profiling to identify specific enzyme deficiencies. For example, elevated levels of ornithine may indicate OTC deficiency, while citrulline elevation levels suggest citrullinemia. So let's start with diagnosis. UCDs are diagnosed through a combination of biochemical tests and genetic analysis. First is biochemical testing, which includes blood tests and urine tests. For blood tests, it is the measurement of ammonia levels that are typically elevated in UCDs. Additional tests may include plasma amino acid profiling to identify abnormal metabolites. And for urine tests, it is the analysis for the presence of specific metabolites that indicate a UCD, such as orotic acid or elevated levels of citrulline and arginine. Second is genetic testing, which includes molecular analysis. Genetic testing can identify mutations in the genes responsible for specific urea cycle enzymes, confirming the diagnosis of a particular UCD. Third is a liver biopsy. In some cases, a liver biopsy may be performed to measure enzyme activity directly and confirm the diagnosis. And lastly, for diagnosis, there is imaging studies. That includes MRI or CT scans. These may be used to check for brain swelling or cerebral edema caused by elevated ammonia levels. Next, we have management. Management strategies for UCDs focus on reducing ammonia levels and preventing hyperammonemic crisis. First is acute management. In cases of severe hyperammonemia, emergency treatments may include: Number 1, hemodialysis. It is a medical procedure used to filter waste products, excess fluids, and toxins from the blood when the kidneys are unable to perform these functions effectively. This is an effective method for rapidly lowering plasma ammonia levels. Second, for acute management, is intravenous ammono fluid. This medication helps reduce ammonia by removing glycine and glutamate from the plasma to maintain hydration and support metabolic function. Number 2 for management is long-term management. We have medications which includes ammonia scavengers or N-carbamylglutamate or carbaglu. First, for medication, we have ammonia scavengers. Medications such as sodium benzoate and phenylbutyrate help remove excess ammonia from the bloodstream by converting it into non-toxic compounds that can be excreted. These agents act as nitrogen sinks by conjugating with glycine and glutamine. Now, how do they work? The sodium benzoate conjugates with glycine to form hippurate, which is excreted in urine, effectively detoxifying ammonia, while the phenylbutyrate conjugates with glutamine to perform phenylacetylglutamine, also excreted in urine, reducing nitrogen load. These work by converting ammonia into non-toxic compounds that can be eliminated. These medications lower blood ammonia levels and alleviate symptoms associated with hyperammonemia. Next for medications is N-carbamylglutamate or carbaglu. This medication can improve ureagenesis, which is the biochemical process by which excess nitrogen is converted into urea, enabling its excretion from the body. In specific enzyme deficiencies like NAG deficiencies and late-onset CPS1 deficiency. We have dietary restrictions. A low-protein diet is essential to minimize ammonia production from protein metabolism. This involves carefully planning to ensure that the diet still provides adequate nutrition for growth and development. Specialized formulas may be used for infants to ensure adequate nutrition without excessive protein. Second is amino acid supplementation. Arginine or citrulline supplements may be recommended as they help enhance the urea cycle function and promote nitrogen disposal. These are to ensure that the body receives essential nutrients necessary for growth and metabolic functions. Next, for long-term management is liver transplantation. In severe cases or those who do not respond adequately to medical arrangement, liver transplantation may be considered. This option is particularly relevant for patients who experience recurrent episodes of hyperammonemia that threaten neurological function where other treatments are insufficient. Timing is crucial. Ideally, transplantation should occur after the child reaches a certain age to minimize surgical risks, but before irreversible urological damage occurs due to hyperammonemia. And last, for clinical relevance, is importance of early detection. Early detection of UCDs is crucial, particularly in newborns who may appear healthy at birth but can develop life-threatening hyperammonemia within days. Universal newborn screening programs can help identify these disorders early, allowing for timely intervention and management to prevent neurological damage.
Moving on, here are the summaries and key takeaways for the urea cycle. The urea cycle is a metabolic pathway in the liver that detoxifies ammonia by converting it to urea. This cycle, also called the Krebs-Henseleit cycle, was pioneered by Hans Krebs and Kurt Henseleit. The general process of the cycle can be summarized simply. Protein breakdown leads to ammonia, which is then converted to urea and then eliminated. But for the specific urea cycle, here's a mnemonic to remember each step. The optimistic clever orange cats always ask for awesome umbrellas. That's T for transamination, O for oxidative deamination, C for carbonyl phosphate, O for ornithine, C for citrulline, A for aspartate, A for argininosuccinate, F for fumarate, A for arginine, and lastly, U for urea. The first two letters T and O occurs before the urea cycle, but we just included it for a more chronological process. Why is the urea cycle so important? First, it converts toxic ammonia into non-toxic urea. It also disposes of two waste products, carbon dioxide and ammonia. It helps form semi-essential amino acid arginine and plays a key role in regulating nitrogen balance. When it comes to nitrogen secretion, urea is crucial. About 95% of it is excreted in urine, 5% in feces, with 80 to 90% of nitrogen lost through urine. To manage toxic levels of ammonia, several medications can help. Lactulose, for example, increases ammonia excretion through the GI tract by causing diarrhea, which acidifies the GI tract to trap ammonium for excretion. Antibiotics like rifaximin and neomycin can reduce ammonia-producing bacteria in the colon, leading to less ammonia production. Other drugs like benzoate, phenylacetate, and phenylbutyrate allow ammonia to react with glutamine or glycine, so it can be excreted in the urine. So, the urea cycle is vital, not just for detoxifying ammonia, but for maintaining our body's nitrogen balance and overall metabolic health.